Hello students, welcome to today's class. Today we will talk on the topic protein and non-protein amino acids. Introduction. A cell is made up of several chemicals. Organic compounds and inorganic matter present in the living organisms are collectively known as biomolecules. Now, depending upon their molecular weight and solubility, they are further categorized into micromolecules and macromolecules. Micromolecules have low molecular weight in between 18 to 800 Dalton. They have higher solubility, simple conformation, and amino acids belong to this category of micromolecules. Amino acids are nitrogenous compounds and constitute the building blocks or the monomers of the protein. Now let us take some examples of macromolecules, and they include proteins, DNA, starch, cellulose, etc. Now over 170 amino acids are currently known to occur in cells and tissues. Of these, 20 different amino acids are constituents of protein and are called as protein amino acids. The remaining ones are the non-protein amino acids. Protein amino acids. Protein form a very important class of biological molecules constituting a major part of animal nutrition. They are the agents of biological specificity and play structural and functional roles in the living organisms. As structural components, they are abundant in muscles, blood cells, bones, and biological membranes. Functional proteins include enzymes, antibodies, and many hormones which carry out roles such as chemical reactions, defensive work, and coordination. Protein on complete hydrolysis yield 20 L-alpha amino acids and are known as protein amino acid. In proteins, the alpha carboxyl group of one amino acid is joined to the alpha amino group of another amino acid by a peptide bond. Many amino acids join by peptide bonds to form polypeptide chain, and each polypeptide chain has an amino N and a carboxyl N. The amino acid sequences of proteins are genetically determined. The sequence of nucleotides in DNA specifies a complementary sequence of nucleotides in RNA, which in turn specifies the amino acid sequence of a protein. Now let us come to the structure of amino acids. All amino acids contain at least one organic acid group, a carboxylic group represented by COH, an amino group NH2, a hydrogen atom attached to a central carbon atom, which is also bonded to a variable hydrocarbon or alkyl group of varying structure, size, electric charge, etc., represented by a R group. The carboxyl carbon of an amino acid is numbered carbon number 1, and the carbon atom to which both COH and the NH2 groups are attached is known as the alpha carbon. Next, we will take up the topic on the stereoisomerism of amino acids. Except in glycine, the alpha carbon in amino acid is asymmetric as it is bonded to 4 different groups. They have been distinguished as D and L amino acids according to their structural relationship to D and L glyceraldehyde. Now next we will see on the classification. How we can classify amino acids. First we will take up on this depending upon their structure and reaction. Amino acids are classified into the following types. Number A, we have the neutral amino acids. These are monoamino and monocarboxylic acids. And some examples are glycine, alanine, leucine, etc. Then in B, we have the acidic amino acids, and these amino acids are monoamino dicarboxylic acid. Example, we have the aspartic acid and the glutamic acid. And in C, we have the basic amino acids, and they are diamino monocarboxylic acids. And examples are arginine, lysine, histidine. And in D, we have the sulfur containing amino acids, and these amino acids sulfur in their structure. And examples is cysteine and methionine. And in E, we have the amidic amino acids, and these groups contain amide groups, and examples are asparagine and glutamine. And in F, we have the aromatic amino acids. They possess cyclic structure with a straight side chain bearing carboxylic and a amino group. Example, we have phenylalanine, tyrosine. And in G, we have heterocyclic amino acids. They have nitrogen in their ring structure. Example, histidine and proline. A more meaningful classification of amino acids we can take up, and that, however, is based on the polarity of the R groups present in their molecules. That is, their tendency to interact with water at biological pH, that is near pH 7. The polarity of the R groups varies widely from totally nonpolar or hydrophobic, that is water-insoluble R groups, to the highly polar or hydrophilic water-soluble R groups. And this system of classification has 4 categories. They are: number 1, we have amino acids with nonpolar R group. The R group in this category of amino acids are hydrocarbon in nature and thus nonpolar and hydrophobic. This group includes amino acids with hydrophobic R groups, and examples are glycine, alanine, valine, leucine, isoleucine, and proline. And in number 2, we have the amino acids with the polar but uncharged R group. Here, the R group of these amino acids are more soluble in water or more hydrophilic because they contain functional groups that form hydrogen bonds with water. And under this category, the amino acids includes serine, threonine, cysteine, asparagine, and glutamine. And in number 3, we have the amino acids with positively charged, also known as the basic R groups. These are diamino monocarboxylic acids. They have significant positive charge at pH 7 due to the presence of extra amino group in aliphatic chain. Example, lysine. And they have a guanido group in arginine and imidazole group in histidine. And in number 4, we have the amino acids with negatively charged, that is acidic R groups. These are monoamino dicarboxylic acids. They have an extra carboxyl group resulting to possession of an additional negative charge at pH 7. Aspartate and glutamate belong to this group. Non-protein amino acids. Besides the 20 amino acids, there are many non-protein amino acids. These amino acids occur both in the free state as well as in combined state, but not in the natural proteins. Many are found as products of intermediary metabolism in the form of intermediates. For example, we have ornithine, L-citrulline, which are involved in the urea cycle. Some are formed by modification of common residues already incorporated into a polypeptide. Homoarginine and diamino titaric acid serve as a source of nitrogen. Beta-alanine is derived from aspartic acid and found in the vitamin pantothenic acid. Gamma-aminobutyric acid, GABA, is found in brain as inhibitory neurotransmitter. Again, we have diaminopimelic acid, which is also an intermediate of lysine synthesis. Then two derivatives of serine, azathioprine and cycloserine, produced by streptomyces, behave as antibiotics. These are two more naturally occurring amino acids which are less common. They are hydroxyproline, found in plant cell wall and in collagen protein, and another is hydroxylysine, also found in collagen. 6N-methyllysine is a constituent of myosin, a contractile protein of muscle. Amino acids show various physical and chemical properties, and they are mainly due to the amino, carboxyl and R functional groups. Let us come to the physical properties. Number 1, we have the amino acids are colorless crystalline solid. Then 2, we have they are generally soluble in water but insoluble in organic solvents like ether. Thirdly, we can say that in neutral aqueous solution, they exist as dipolar. That is, they have a dual nature of acidic and basic groups, which are contributed by carboxyl group, amino group, and are known as the ampholites. And fourthly, they show deuterionic nature of amino acids. That is, amino acids are amphoteric compounds. That is, they have ionizable properties. The carboxyl group is acidic and can donate a proton by dissociation. The amino group, being basic, can accept a proton. When the proton gets dissociated from the carboxyl group, it is attached to the amino group and the amino acid is said to be deuterion. Important chemical properties shown by the amino acids. Chemical reactions or the chemical properties of amino acids are due to the carboxyl group, the amino group and the functional group of the side chain represented by R. Now, first we will discuss on the various reactions of amino acid due to the amino group. Number A, N-alkylation and N-arylation reaction. Here, the two hydrogen atoms of NH2 can be replaced by aliphatic radicals such as methyl and obtain N-methyl, N-dimethyl or N-trimethyl derivative by treatment with methyl iodide in alkaline solution. In arylation reaction, the two hydrogen atoms of NH2 can take place to obtain aromatic radicals like dinitrophenyl, represented by DNP derivatives, which is used for determining the N-terminal amino acid of proteins. And in B, we will see the N-acylation reactions. Here, the amino group of the amino acid can be acylated with formyl, acetyl or carbobenzoyl radicals by treatment with acid anhydrides or chlorides under proper condition. In vivo, that is inside the cell, N-acetyl and benzoyl derivatives of glycine is formed during detoxification process. N-formyl methionine plays an important part in the initiation of protein biosynthesis in bacteria and organelles. These are the examples of acylation. Number C, we will discuss on the Sanger's reaction. This reaction was introduced by the scientist Frederick Sanger, who used 1-fluoro-2,4-dinitrobenzene FNDB. This reaction is specific for the free amino group of the amino acid or polypeptides, resulting in the formation of DNP derivative, a yellow crystalline compound. In D, we have the Edmonds reaction. It is a useful reaction employed to identify the amino acid at the end terminal of a polypeptide chain and is hence used in the determination of amino acid sequence. The reagent phenyl isothiocyanate reacts with the amino acid to yield a cyclic product known as phenyl hydantoin, which proceeds in the following steps. Then next, we have the densyl chloride reaction. Here, the amino group of amino acids and proteins react with the densyl chloride to produce a densyl derivative with a fluorescent property. By this method, even the presence of a small trace of amino acid can be detected. And lastly, we have the action of nitrous acid. Nitrous acid reacts with the NH2 group to form the corresponding hydroxyl acid, liberating nitrogen, which can be titrated. This is the principle of Van Slyke method for the determination of the free amino group of amino acids, peptides or proteins. Now, let us see or discuss on the various reactions which can take up due to the presence of the carboxyl group. In A, we will see formation of salts. Amino acids form salts when titrated with base, that is, we can take an example of NaOH in alcoholic solution. This property may be used for titrating amino acids, but due to the weak dissociation of the carboxyl group, high pH value of 11 to 12 must be reached to obtain total saturation. And B, we have the decarboxylation. Decarboxylation can be carried out in the laboratory by heating, preferably in the presence of barium hydroxide. The carboxyl group of alpha amino acid may also undergo enzymatic decarboxylation to yield a corresponding amine in the large intestine. Biological amines are important compounds which participate in various physiological reactions. Some examples are histamine, tyramine, tryptamine, etc. Then in the C, we have the acylation reaction. The carboxyl group of amino acid can be acylated when treated with phosphorus pentachloride PCl5. In D, we have the esterification. The carboxyl group of amino acid can be esterified with an alcohol in the presence of dry HCl to form the corresponding esters. Amino acids can also undergo deamination and transamination reaction, causing the removal of the amino group. The Ninhydrin reactions shown by the amino acid. Ninhydrin is a strong oxidizing agent which is able to carry out oxidative decarboxylation of amino acid to yield carbon dioxide, ammonia and aldehyde and a reduced form of ninhydrin. The amino acid is first transformed into an amino acid which is further hydrolyzed into an alpha keto acid and then gets decarboxylated into one carbon atom less aldehyde. Identification of the obtained aldehyde determine the amino acid. The reduced ninhydrin reacts with additional molecules of ninhydrin in the presence of ammonia produced in the first reaction to yield a blue-purple colored product which is quite specific, showing an absorption in the vicinity of 570 nanometer. This reaction is useful for the estimation of amino acids quantitatively with the help of a spectrophotometer. Today's discussion is in the form of conclusion in this way. Amino acids, which are the building blocks of proteins, are 20 in number. Generally, L form of the amino acids occur in natural proteins. Other less common amino acids also occur in either as free metabolites, intermediates, or constituents of few proteins like bacterial, cell wall, and muscle protein. They show various physical and chemical properties too. Their chemical properties are due to their carboxylic, amino, and functional group of the R side chain.